Hi, I'm Harry Rathbone and I'm a PhD student in Biophysics. You may not think that biology and physics have much overlap, but life too must obey the laws of physics. Laws which, in this case, are subtle and intricate, owing to the complexity of life itself. A physicist's view of life breaks down biology into flows of energy and matter, resource use, and flows of information, all in service of self-replication and keeping the cell out of equilibrium with its environment. My work focuses on how energy is transported and converted in cells during photosynthesis. I work on a peculiar group of photosynthetic organisms called cryptophytes, which have evolved specialized organelles packed with light-harvesting proteins. Proteins are massive, complex molecules that take a certain shape to perform a certain function in the cell. Essentially, inside these cryptophytes is a bag of proteins that harvest and transport light. Light energy is transported by hopping between molecules until it reaches the reaction center where it is used. Cryptophytes live in deep marine environments where light is scarce, so harvesting energy with the highest efficiency is paramount. The photosynthesis process begins by the absorption of a photon by dye molecules embedded in proteins. Upon absorption, an electron is promoted to a higher energy level, and that electron and the hole it leaves behind form a bound state called an exciton, or a particle of excitation. This state has a lifetime on the order of nanoseconds, so energy transport has to be very fast. Energy transport is done by the exciton hopping from molecule to molecule until it finds the reaction center where it is used. The exciton on one molecule produces an electric field which then interacts with a neighboring molecule. They then electronically couple and the exciton can hop across. If the lengths between molecules were any longer, they would have to employ a photon to transfer energy away. The main focus of my research is to see how quantum coherence is used in organisms to increase energy transfer efficiency. In the quantum world, particles behave like waves and can be in multiple places at once. In this setting, coherence means the exciton can be spread across a number of molecules at once. Unfortunately, cells are hot, wet environments, and all of this thermal noise destroys any coherent state that can be set up. However, if two molecules are so close together they almost touch, a coherent state can be set up that lasts a lot longer, which can increase energy transfer efficiency to the next molecule. This entire process is a very specific example of an interfering quantum warp, which is expected to be employed in quantum search algorithms in quantum computing. On paper, this works out beautifully. However, my task is to see whether this is biologically relevant, if it's under biological control, and if this process really increases energy transfer efficiency in the cell. We study these organisms because A, they're an interesting quirk of nature, B, because they help us understand how biological systems have evolved to have the greatest efficiency at resource use, and C, because they have potential applications in increasing solar cell efficiency and other applications potentially in quantum computing. To analyze proteins in cryptophyte algae, we use a variety of different techniques. One tried and true technique from the time of Newton is absorption and emission spectroscopy. In this technique, a sample is illuminated by a specific band of frequencies, and then the emission spectra is measured. The difference between the in and out spectra gives you the absorption spectra, which shows absorption features and fluorescence features. This allows us to isolate frequencies and therefore, by Planck's law, energies of interest. In reality, these dye molecules have very complex spectra. If the energy of the photon is greater or less than the energy that it takes to excite an electron, then rotational and vibrational transitions can also be excited or de-excited. A key feature of proteins is that they have, more often than not, a definite folded state, barring some diseases which cause misfolded proteins. To study the folded state of proteins, we use a process called circular dichroism spectroscopy. Proteins are complex, curled up molecules with some definite structural features, such as alpha helices, beta sheets, and random coil. CD spectroscopy uses left and right hand circularly polarized light and measures the ratio of absorbance between the two. You'd expect that circularly polarized light would interact with structures that have different circular symmetries. This ratio tells you about the proportions of structural features within the protein. Almost all helices found in nature are right-handed, barring some examples in bacteria where left-handed helices are used for toxins. So it's often easy to see the folded state of the protein based on this absorption. The usefulness of this method arises when we're looking at protein folding in different environments, such as changes in pH. Part of our analysis involves looking at the structures of proteins, which correlates to their function and physics. And in my project, we can look to see where dye molecules are placed within the protein. A classic method to do this is X-ray crystallography. This technique uses high-energy X-rays, which then diffract through the crystallized proteins. Each protein forms a cell in the crystal, which tessellates to form a regular pattern. The atom in each protein is therefore regularly spaced in the crystal. X-rays then scatter off the regular lattice, which creates a diffraction pattern. Diffraction patterns can then be analysed to give us spatial information about the atoms in the proteins. We take an image of the diffraction pattern from every angle around the crystal, which allows us to generate a 3D structure of the protein. Pretty much all images of proteins are produced this way. I got interested in physics from a very young age. Growing up on a farm in the outback, I had a phenomenal view of the sky, which is why I was originally taken by cosmology. However, I've recently made the move to biophysics in my honours year, having learnt that we can understand life through the physics that I so loved, and I have not looked back since.